Promise. Okay. Slideshow. Play from the start. So this one, we go over the ecdytozoans and the leucotrophozoans. These are all triploblastic, right? We're advanced on from all these crappy animals up here to the triploblastic animals. Okay. So this group, or this lecture specifically refers to the protostomes. How do the protostomes differ from the deuterozoans again? Give me the three things. So the first pore turns into a mouth in the protostomes versus an anus in the deuterozomes. Number two, how do they split up? Spiral versus radial cleavage. And finally, what about the kinds of the cells that are in that 8-cell embryo? Terminate versus indeterminate. Perfect. You guys are... I will say that this lab section specifically, I think, got like 5 points above average on this last lab practical, which is insane. So maybe I'm lecturing the wrong group, but so that's what I'm talking about. For the other classes, yeah, but not for this class. All right. So for the protostomes, there's just two groups you got to know: the ecdytozoans and the leucotrophozoans. How do they differentiate from each other? Exoskeleton molting. That's the one thing that you got to know, right? That's how they are joined together in these two clades. And again, these are the clades that don't actually have a phyla or a class, which is tricky, but you will have to know the terminology for that. So leucotrophozoans, ecdytozoans, those are the things you got to know, even though they're not assigned to a clade or a superphyla or whatever like that. Okay, so they're all bilaterally symmetrical. They're in this group called the bilateria, right? Except, of course, the sea urchins. And we went over all this bullshit. So just remember, for radial cleavage, for spiral cleavage, just look at this 8-cell embryo. If this line is not right above this line, it's spiral cleaved. Okay. Okay. This is going to be really, really important. We already talked about the germ cell layers, and we already talked about a little bit about the coelom, right? Just one more time, for my benefit, what is the purpose of a coelom? It's this body cavity inside of you. What is the purpose of it? Protect your organs, right? An airbag and gas exchange. Yep, perfect. Three kinds of triploblastic animal. Either acoelomatic, which is the endoderm is connected to the mesoderm, which is connected to ectoderm, in the flat room over here. Or a pseudocoelom, which has no mesoderm around the gut. So that's what distinguishes a pseudocoelom from a true coelom. Remember that. No muscle around, or no mesoderm around the endoderm. Okay? And why is that important? What makes this more advanced than this? What does having muscle, or what does having mesoderm around your internal organs do? Two things. Better digestion, one of those things, right? So the first, let me just go back real quick. The pseudocoelom, the only organism that you need to know that has pseudocoelom is the nematodes. That's the only one, okay? So it's a perfect internal comparison between two worm-like creatures. The nematode has no mesoderm around the endoderm, which means there's no muscle around the gut, which means in order for them to poop, they have to move around, right? To move food through their body, they have to move around. But for us, our muscles on the inside, they can just pulsate our digestive tract, which can move food through it. Yep. Secondly, mesoderm isn't just muscle, it's also connective tissue. So you can see in the true coelomatic organisms, they have these connections from the outer mesoderm to the inner mesoderm. Which that does is it takes your internal organs and anchors them to your external body. So it gives it's like having an anchor. It's like if I were, what am I trying to say? So if you're like Spider-Man sitting around on the inside of a room, you shoot a bunch of webs and then you're sitting there like a spider, right? You can move around like this, but you're still anchored to the outside, right? Versus if you're just Spider-Man in a room without the webs, the outside rooms you hit the walls and shit, right? It's not. So you get some amount of plasticity, but still structure. Okay. So anchoring to the walls of the cavity and muscular movements of your internal organs. Cool. Awesome. All right, let's go into the locotroph zones. So you have two groups you need to know: the annelids, which are the segmented worms, and the mollusks. Okay. For the annelids, there's just a couple of things you want to know, I guess. So how they differentiate from each other. Let's not focus on how they differentiate from. Just remember the different biological traits of each individual group. So the annelids, they have these trochophores. These are both in mollusks and annelids. That's their larvae. A lot of them have these guys, which are these larvae that have cilia all around them that look like that. They're not really that important to know except to memorize that name, right? They both have true coeloms. Remember, all these triplets have true coeloms except the nematodes. Metamerisms, what are those? So at this point in time, these are just vocabulary words you just got to memorize. What are metamerisms? They're segments, right? And what is the purpose of having a segment? Redundancy, right? Each metamer in a earthworm specifically, we'll focus on the earthworms, they have kidneys and they have motile structures. So they have arms and they have kidneys. So if you cut one segment off of that worm, you will still be able to filter your blood and you can still move, right? Parapoids, which are little external parts of their hydrostatic skeleton, these little fake feet that like move around like that. Not present in earthworms, or very reduced in earthworms. Hydrostatic skeleton, remember, 3 groups of animals that have hydrostatic skeletons you gotta remember. 4, sorry. The 2 worms, the nematodes, and the earthworms, right? The annelids, the nematodes, the echinoderms, and the jellyfish, the cnidarians. Remember those groups. Remember that they have hydrostatic skeletons. These guys are monoecious usually, which means they have both male and female organs in the same group. Okay. So we'll focus on that for now because that's the next section. I don't really know what I'm going to ask Alice, and I'll get back to you guys on how much you're expected to know for the dissections. Because really, what they told us to teach you was to cut it open and just, this is that, this is that, this is that, this is that. They didn't really give us like a nice broad purpose for like how that function in a evolutionary sense. So I'll ask her and I'll get back to you, okay? But just go into the notes and go into the front of your lab notebook and look up all these different structures and what they do. That's really all I can tell you there. I will have notes that kind of highlight that, so don't worry about that too much. But theoretically, if like Alice wanted to be an asshole, she could like screw you guys over with this kind of stuff. So that's what I'm saying. I don't really know, but again, I'll let you know. Okay, so forget about that for now. All right, mollusks, their big thing. Sorry, let me go back. Just some quick features that you probably should know, though. The setae are those hair-like features that are on the outside. I don't know. I'll put these in the notes. Never forget about this. All right, the mollusks do have a lot more very specific features that you've got to know in evolutionary context. Number 1, they all start out with shells. Their ancestor have shells, but they don't always have shells, right? The ctenidia. Remember what the ctenidia are. The ctenidia are the gills, yeah? And the gills are in different parts of different mollusks, and they function for different reasons. So the things I would definitely remember are all on this slide. These are things that kind of put all mollusks together. Head foot, which is muscular, like this muscular fleshy lobe. It's either the foot right here, or it can be this thing right here. It's just wherever the mollusk has one big muscular part. In the cephalopods, their foot becomes like this section and this section, so it's not really that important. But for most of the mollusks, so it's good to have the basic mollusk structure down pat. You have this fleshy foot, you have a shell. Underneath the shell is the mantle. The mantle is the covering for all the internal organs, right? And inside this mantle, there's this cavity right here. It's one open-ended cavity which has the ctenidium. So remember, the mantle cavity is the cavity that holds the gills. It's got a mouth with a radula in it, which is a bony tongue that rasps away at stuff. The mantle covers all the internal organs, and the shell covers the mantle, if it's there. Okay, so let's talk about how these individual groups are different. So we have 4 classes that you need to remember. Number 1, Gastropoda. Those are the snails. How they differentiate from everything else. They have a very well-developed radula, so they can rasp away at stuff. Their GI tract is kind of curved in on itself, so the tenidia in the mantle cavity is actually right above the head. And the anus actually comes into the cavity, so it poops out of its lungs, kind of. Yeah, it's gross. It's got a big foot, which allows it to move around on the surface of the thing. And you'll always want to reference that back. If you take pictures of the individual mollusks, anything with this big fat foot is going to be a gastropod, except these guys. So there's a possibility of fucking up the identification between the gastropods and the polyplacophores. But what is different about the chitons is they have multiple shells on top. Okay, they have these little flat shells on top like that, and that's very, very obvious. So if you look at it from the bottom, these guys might look the same, anything with a big flat foot, but these guys have multiple shells on top. Remember that. Okay. Also, so those shells let you they let them roll around in a ball. They have a really fine-tuned radula, again a big ass radula. How you can tell this apart besides just the little different shells? They have gills lining the sides of their mantle. So they don't have a specialized mantle cavity like these other guys. They just have gills like right here, just all along the sides of their foot. Okay? The cephalopods are very, very different from everything else. They're very highly derived mollusks. They have very well-developed eyes. They have a siphon. Remember, there's a siphon that shoots out the water, which also contains the ctenidium. Most of them do not have shells. If they do have shells, they're the nautilus, the nautili. If they don't have shells, they're either greatly reduced as this little pen inside, or a cuttle bone like the cuttlefish, or in this case of the octopi, they have absolutely no shell whatsoever. So remember, ink sac, they have a beak to chomp on stuff, a siphon to move around, and tentacles, and no shell. All right. The bivalves, so what you need to know is that the ctenidia are used as what in the bivalves? Their lungs aren't just used as lungs, they're used as filter feeding appendages, right? So they have a siphon. They usually have a siphon coming in, going back out, and the water runs through the gills. The gills will take in. The gills are actually called lamellae. They have another term, not just ctenidia, that's called lamellae. Just more shit for you to remember. And they have 2 shells. Anything that has 2 shells and looks like it has 2 shells is going to be bivalve. Okay. There are a lot of other small morphological features like the abductor muscle, stuff like that, but that's all in your lab notebook, so you can remember that. The major takeaways are that it uses its ctenidia, so it doesn't have any mouth parts. It uses ctenidia to capture food, as gills, but also to capture food, and ctenidia are called lamellae. As far as identification, there's only one thing that you might be able to fuck this up with. They're the barnacles. And if you look at the barnacles are actually arthropods. They're what they are they're basically lobsters that go backwards in their shells. But if it opens up and it has things that go out like that, that's going to be a barnacle, and it's not a mollusk. Okay. Everything else that has 2 shells is going to be this, a bivalve. Okay. Great. So remember all these different structures, remember them, and make sure you can identify them in each one of these different groups. Again, just look at your notes, look at the lab manual. Yeah. All right, this last group, the final thing we're going to talk about today are the ecdytozoans. These guys will shed their skeletons. Okay? Actually, real quick, let's go back to the squid. I think the takeaway of this specific dissection is that it's got that reduced pen, right? So it used to have a shell, but it doesn't have a shell anymore. Make sure you know the beak. These are the ctenidia, the gills. It's got an ink sac. Yeah, I guess that's it. All right, so these guys shed their exoskeletons. You have the nematodes. Let's go over the nematodes real quick. Nematodes, as far as you need to know, they're very, very simple. They're just a sac full of gonads and GI tract. Very, very simple. Okay? And the reason they're so simple is because most of them are parasites. They live in your gut, so they eat the stuff that you already digested. And then they poop out eggs into your poop, and that's how the eggs get released. So very, very simple. Nothing you really need to know about these guys except they are pseudocoelomatic and they have a hydrostatic skeleton. That's it. Okay. Arthropods, a lot more complicated. They are your millipedes, your centipedes, your scorpions, spiders and insects, everything that kind of looks like that. Crustaceans, right? They have a cuticle, a very thick exoskeleton made out of chitin and other stuff, but mostly chitin. These are true coelomatic. They have joint appendages. So things I would know, each segment, each group, they have all these segments, but in the more advanced ones, they have segments that group together to make larger segments. A lot of small segments turn into a large segment, and they specialize into different features, right? So you'll have like an insect, which will have an abdomen, a thorax, and a head. The head is all those head segments come together, and they all differentiate from each other to turn into like eyes and like fucking like antennae and stuff like that. So they're all the head is all for processing of sensory stuff. The middle is the thorax, and all those segments turn into things with specialized legs and wings and shit like that. So it's for locomotion. And finally, the ass, the abdomen has respiratory features. It has digestive features, and it has sexual features. Remember in the arthropods, so you can either have 2 tagmata or you can have 3. The 3 are the insects: abdomen, thorax, and head. For all the other guys that have 2, the spiders, the arthropods, so on and so forth, the first segment is the cephalothorax. So it takes both the purpose of sensory stuff and for moving around, right? It's got the thorax and the head, cephalothorax. And then you got the abdomen. Okay. What do you need about these guys? Really, I don't think there's that much to know except a couple of things. Number one, be sure to identify the different groups. So everything that has a hard skeleton that looks like a bug, any kind of bug, like a millipede or a scorpion, so on and so forth, that is going to be an arthropod. The only thing that you might be able to screw up on, again, is the barnacle. Go home, look at the pictures of the barnacles. Please look at barnacles. I didn't take pictures of the barnacles this time around, but does anyone have lab photos? Oh, shit. Okay, I'll post pictures of barnacles because those you might fuck up on because they kind of look like clams. They really do look like clams, but they're not. They're basically this guy, but in a shell, and its ass is hanging outwards. And so if you see like if you see something that looks like a clam, but inside the clam there's stuff that's going out like that, those are basically these things in the crustacean. These are crustaceans. Know that dude, I lost my train of thought. What are these barnacles? Know that the barnacles are crustaceans. So there's 2 kinds of barnacles. There's these ones that open up like that, and there's another one that looks like a volcano that doesn't have 2 shells. It's 1 shell that opens up like that. Just remember that. Okay. Things maybe to remember about the crayfish dissection. Like they tell you to know all these different like pieces of the tail, but they don't do anything differently. They really don't. Like a telson is not that different from your pod. It's so like in the last gasp, when you like are done with everything else, all the conceptual stuff, maybe start memorizing all these different parts. But otherwise, it's not really that important. Know that the swimmerettes, they look different because the males don't have big ones. The females have big ones because they need to aerate their eggs. That's where they keep their eggs. And then also know that the gills are attached to the legs. Right? Remember that when we pull them apart, the gills are attached to the legs. So when it's moving around, that's when it's actually breathing. Like well. Other than that, I will ask people that have taught this class before, and I will get back to you on how much detail you really need to know for the dissections, because that's really what's holding this lecture back. Otherwise, I think it makes perfect sense. All right, class dismissed.